Toxins. Bacterial toxins. The bacterium produces a substance that is toxic to human cells. The toxin causes the symptoms of diseases. We consider examples of each of the different types of bacterial toxins. Non-protein toxins. Peptide and protein toxins. Non-protein toxins, also called as endotoxins. Endotoxins include lipopolysaccharides LPs, lipodecoic acid LTA, and peptidoglycan PG, which are components of bacterial cell walls. LPs, found in the outer membrane of gram-negative bacteria like Escherichia coli, leading to the production of pro-inflammatory cytokines. This response can escalate to septic shock. For example, Bordetella pertussis, the causative agent of whooping cough, releases a form of PG called tracheal cytotoxin TCT. TCT disrupts the respiratory system by arresting ciliary movement in the respiratory tract, impeding the clearance of mucus and leading to the characteristic severe coughing fits of pertussis. Protein toxins, also called as exotoxin. They are classified into three main types based on their mechanisms of action: Type 1, Type 2, and Type 3. Type 1, superantigens. Type 2, membrane-disrupting toxins. Type 3, AB toxins. Superantigen, X, gram-positive S, aureus. These SAGs are not processed by proteolytic digestion inside macrophages, but bind direct to MCHE2 on the macrophage's surface, leading to secretion of high-level IL-2, cause shock symptoms. Membrane-disrupting toxins. Example, Listerialis O. Pore-forming toxins create pores in the phagosomal membrane to escape from phagosome into the host cell cytoplasm. Example of cytolytic enzymes, phospholipids, hemolysin, cytolysine. Instead of creating pores, they enzymatically degrade membrane lipids and other molecules, such as hemolysin produced by E. coli and S. aureus. In AB toxins, the A, which is active component, has enzymatic activity, causing toxic effects within the host cell, while the B binding component targets and binds to specific host cell receptors. The B component facilitates the delivery of the A component into the cell, allowing it to disrupt essential cellular functions. The diphtheria toxin, made by Corynebacterium diphtheriae, blocks cells from making proteins by changing a key protein called EF2. This toxin adds a chemical group called ADP-ribose to EF2 in a process called ADP-ribosylation. This change stops EF2 from working, so the cell can't make the proteins it needs to survive, leading to cell death. The botulinum toxin, produced by Clostridium botulinum, is a neurotoxin that blocks the release of acetylcholine at neuromuscular junctions. Without acetylcholine, muscles can't contract, leading to flaccid paralysis, where muscles become weak and limp. Effector proteins are directly secreted from the bacterial cytoplasm and injected into the host cell's cytoplasm through specialized secretion system, similar to type 3. The primary difference is that toxic effector proteins are non-toxic by themselves. They need specialized secretion system to deliver them into host cell in order to exhibit their toxicity. Examples of toxin-mediated disease. Diphtheria toxin DT is one of the best studied bacteria toxins. Here is the summary about it. Corynebacterium diphtheriae causes diphtheria. It is transmitted via aerosols and colonizes the throat. As the bacteria move down the throat, they form a pseudomembrane, which is a thick, grayish layer that can bleed if removed. DT production is influenced by iron availability. Production increases in low iron conditions as a regulatory adaptation. Lysogenic Corynebacter IFS bacteriophages that integrate their genome into bacterial DNA carry the gene for DT, meaning only C. Diphtheriae infected with these phages produce the toxin. DD binds to heparin-binding EGF-like growth factor, HB-EGF on host cells, facilitating its entry and toxic action. Mechanism of action of diphtheria toxin. It functions as an A and B toxin, where the B component binds to host cell receptors HB, EGF, allowing the A component to enter the cell. Inside the cell, the A component inhibits protein synthesis by ADP ribosylating elongation factor 2EF2, leading to cell death. Diphtheria antitoxin. Antitoxin derived from horse antibodies was historically used to treat diphtheria before antibiotics were available. Diphtheria vaccine. The vaccine uses a toxoid, which is a modified form of the toxin that is no longer toxic but still stimulates an immune response. Toxoids are key to vaccination as they allow the body to develop immunity without the risk of toxin-induced disease. Clostridium botulinum produces BONT, which is a heat-sensitive toxin that leads to paralysis. It goes straight for nerve endings at muscles and blocks acetylcholine, which is the chemical that muscles need to contract. Without acetylcholine, muscles become limp and cannot move. Unlike other toxins that kill cells, BONT does not destroy them, so recovery is possible, though it is a slow process as the body rebuilds those nerve connections. Mechanism of botulinum neurotoxin. BONT cleaves snare proteins, neuron-specific proteins involved in neurotransmitter release, preventing vesicles containing acetylcholine from fusing with the cell membrane to release acetylcholine into the synapse. Clostridium tetani produces tetanus toxin, which targets snare proteins like botulinum neurotoxin does. Tetanus toxin binds at neuromuscular junctions but acts in the central nervous system, blocking inhibitory neurotransmitters gamma-aminobutyric acid and glycine. This causes spastic paralysis, or lockjaw, where muscles stay continuously contracted. In some local infections, tetanus toxin can also cause local flaccid paralysis. Botulinum neurotoxin acts on peripheral neurons and causes flaccid paralysis, while tetanus toxin targets the central nervous system, leading to spastic paralysis. Both toxins cleave neuron-specific snare proteins, but botulism affects acetylcholine and causes muscles to relax, whereas tetanus blocks GABA and glycine, resulting in constant muscle contraction.